Alzheimer's disease is the leading cause of dementia in older adults and the seventh leading cause of death in the United States. We currently don't know what Alzheimer's disease is at the cellular level. A project called the Seattle Alzheimer's Disease Brain Cell Atlas, or CAD for short, is focused on answering this exact question. This type of research is considered fundamental or basic science. Before we can develop treatments or cures, we first need to understand the underlying biology. The project has three main questions. First, which brain cell types are affected in Alzheimer's disease? Second, when are these cell types affected during disease progression? And third, where are these affected cell types in the brain? How did they answer these questions? The first step was to find postmortem brain tissue with Alzheimer's disease. In the Seattle area, 84 participants agreed to donate their brains for neuroscience research once they passed away. These donors represented the full spectrum of Alzheimer's disease severity. Finding these participants was done in partnership with Kaiser Permanente and the University of Washington. Then researchers measured three types of data from these donated brains. In neuropathology, they looked at abnormal protein aggregation. With genomics, they measured RNA at the single cell level. With spatial transcriptomics, they measured RNA of individual cells and their spatial locations within the brain. We will briefly go through each of these measurements. Let's start with neuropathology. The hallmark of Alzheimer's disease is abnormal protein buildup, specifically amyloid beta plaques and tau tangles. This is often measured as a binary, yes or no for protein aggregation. In this project, the researchers used machine learning to gain a deeper understanding of the specific levels of protein aggregation. Now, instead of a binary yes or no, individual brains are assessed on a continuous scale, spanning from no protein buildup to high levels of aggregation. Now, with the quantitative neuropathology data, the researchers created a model to categorize how severe Alzheimer's disease was for each of the donors at their time of death. This was a way to estimate how far the disease had progressed for each individual. This model was critical to map all the collected data to the level of disease severity. The next major data source was single-cell transcriptomics. This technique allows us to measure an individual cell's gene expression, which we can then use to define the cell and what type of cell it is. Before this Alzheimer's project, a previous project was done through the Brain Initiative Cell Census Network, where cell types were defined in the same area of the healthy human brain using single-cell transcriptomics. By using these data from the healthy brains, researchers could compare and see differences at the cellular level with the Alzheimer's brains. The main findings were there were no new neuronal types in the Alzheimer's brains. However, there were new non-neuronal types in the Alzheimer's brains. They also found that both excitatory and inhibitory neurons decreased their abundance, and that the abundance of microglia increased. Lastly, they saw that the abundance of oligodendrocytes decreased as well. Although inhibitory neurons decreased overall, there is a specific subtype of inhibitory interneuron called somatostatin, or SST, and this subtype decreased very early in disease progression before all the other neurons. These neurons died off even before the hallmark plaques or tangles formed. This was a novel finding that was only made possible due to the combination of single-cell RNA sequencing and quantitative neuropathology. Why was this specific inhibitory neuron affected first? Does the death of this neuron type cause other neurons to die after? We don't know. More research is needed. The last major data source was spatial transcriptomics. Spatial transcriptomics is an emerging technology developed in the early 2020s to measure gene expression of individual cells while maintaining their spatial location within a sample of brain tissue. In previous single-cell transcriptomic technology, we actually lost spatial data because the cells are individually extracted from the tissue sample. As of 2026, there is no spatial transcriptomic dataset of the entire human brain, healthy or unhealthy, but this is something that many researchers are actively working on. In this specific Alzheimer's study, tissue samples from the middle temporal gyrus were analyzed for spatial transcriptomics. The middle temporal gyrus is affected in Alzheimer's disease and is known as a transition zone in disease progression. Using spatial transcriptomics data, we can locate where exactly those affected SST cell types are located within the brain. It turns out that a subtype of SST cells, specifically in layers 2, 3, and layer 4 of the cortex, were most vulnerable to Alzheimer's disease. Different layers of the cortex have different cell types. In addition, they also have different short-range and long-range connections. SST cells inhibit other cells, both excitatory and inhibitory. Since the layer 2-3 SST cells died so much earlier compared to the layer 2-3 excitatory cells that they inhibit, this suggests that Alzheimer's disease is a circuit disorder. The Seattle Alzheimer's Disease Brain Cell Atlas project was the first attempt to characterize Alzheimer's at the cellular level. There is still a lot of work to be done, such as recruiting a more diverse cohort, measuring more genes, and conducting spatial transcriptomics on more areas of the brain, as well as analyzing blood samples from living patients, and more. This atlas is a powerful first step towards truly understanding what Alzheimer's disease is and how exactly it affects the brain at the cellular level. Now that you understand the kinds of data that were collected for this project and some of the findings, I invite you to explore the data for yourself. Links to all the open data are available in this video description.